Thank you so much, Dave. Thank you all for allowing me to share with you. I wish I was there with all of you, but I'm grateful for this opportunity. So, what my topic is here for us at the moment is um, emphasizing and showcasing the tools that we have to be able to characterize black spot resistance in roses, and in particular, using detached leaf assays or evaluations. And we've been using these different kinds of assays here for a long time at the University of Minnesota. So years ago, when Vance Whitaker was a master's student with Stan Hokanson, the focus began in terms of better understanding resistance to black spot. So Kathy Zuzak was breeding roses there at University of Minnesota, and one of the challenges was always roses may look really good for resistance for first couple years, and then suddenly get taken down heavily by black spot. So that started the focus on black spot resistance at the University of Minnesota. And when Vance started, um, he looked through the literature. He saw that there were different reports on black spot resistance in the United States and Canada, and they isolated different forms of Diplocarpon rosea, the fungus that causes black spot, and characterized them into different physiological races. But unfortunately, those isolates were lost and weren't available any longer. So he recollected different isolates, worked with people that shared things with him, and that started the work. And one of the things, I'll jump ahead here, that Vance focused on as part of his studies was looking at differences in partial resistance or quantitative resistance in some different populations. And during that study, he compared doing whole plant inoculations with detached leaf assays. And we found that the correlation was pretty high. What we saw in the detached leaf assays was pretty comparable to whole plant inoculations. And whole plant inoculations take a lot of resources. So we're propagating these roses, getting them to a good size, having a lot of replicates, and then in a situation like this in the greenhouse, creating an environment, typically working with just one physiological race to be able to unify at least the form of the pathogen that we're working with at the moment. So these detached leaf assays from this point on have been the focus in the program. So what advantages are there to detaching those leaves and doing those inoculations in that controlled setting, typically in the lab? Well, we can use just a specific isolate, have a particular race, and that makes controlling that pathogen a lot more easy. In addition, we can leverage a limited number of plants that we have growing in the greenhouse where things are free of black spot and harvest those leaves, bring them into the lab cycle after cycle, week after week, to be able to eventually get through the characterizations for that cultivar across many different isolates or races. So in the greenhouse, we typically don't have any black spot because black spot needs about 6 or 8 hours of the spores on a wet leaf in order for the spore to germinate and penetrate. So as we're watering plants in the greenhouse, aiming the water to the soil, we just don't have that continuous moisture on the leaves. So that's a wonderful situation. And then in these controlled environments, these boxes where humidity can be held relatively high, and we could put a specific isolate of black spot on leaves of a particular host, we can be able to get that control that's necessary to find those roses that are resistant to certain races and those that are susceptible. And Dave just mentioned a little bit about that vertical or qualitative resistance conferred by RDR genes. All right, so here's a picture of roses in the greenhouse just a couple weeks ago. So lots of leaves. So we could harvest leaves over time, get enough leaves for replicates, enough boxes to do lots of different isolates. So we want to avoid powdery mildew. So traditionally, what we would do is burn sulfur at night, and the sulfur from our studies and stuff in the literature doesn't seem to affect black spot. So that's great. So a little bit of burning of sulfur allowed us to not have a powdery mildew issue. We've been banned from using sulfur in terms of burning it at night recently in Minnesota. So um, we found a couple of fungicides that are very powdery mildew specific, but we only use them if and when we need them. So we've been trying to control the environment enough with humidity, lack of water stress of the plants to overcome that challenge. So when Vance started to do detached leaf assays, the question that came to the forefront is, what kind of container do we use? What can be affordable, abundant, easy enough to work with to use for the detached leaf assays? So at that time, I was doing some potato tissue culture, and I was using these food grade salad containers that come sterile. They're irradiated from the food industry, and they were a good size, 9 inches by a little over 7, a little over 2 inches in height, and they were perfect. They're polystyrene. We're able to wash them out, use them, which is great. One of the challenges recently here is that after over 25 years of this particular model from this company, Dart, they discontinued it. So, and they don't have something with comparable dimensions, unfortunately. So we've been very careful to wash out and reuse these. We bought a few boxes here at the bitter end before all of them were sold. And so doing some sleuthing, there's an additional company here called Start Packaging that has a corn-based plastic version that's actually the same size as that Dart brand. So we got some, we tried it out. The only difference is that they say that it just can't handle high temperatures, and we're not cooking anything in it using high temperatures. So if anybody wants to pick up and do some of these assays, here's an alternative that we could work with. So here's a washed out, used multiple time polystyrene version on the left, and there's that new corn plastic version that we got some samples of on the right. And the lids here, I have a lid perpendicular just sitting on the top, are the same size, they're interchangeable, they fit. So we're grateful for this resource. So in order to hold the moisture within this container, having some moist paper towel does the trick. And using the Bounty Selecticides is perfect. It's just the right width for a sheet, and we fold over an end of it so it can fit in the box in that 9 by 7 inch container. So we want to moisten the paper towels. So we've learned that these little beakers that fit 30 mils work perfectly, and we use distilled water. So we just pour it in there. It wicks to the edge, which is great. And then now as we conduct those detached leaf assays and acquire the leaves, we have to be very careful. So we want to use leaves of consistent firmness, age roughly, in order to have consistency across our reactions. We know that younger leaves tend to be more susceptible than older leaves, which is really interesting. In the landscape, it's the oldest leaves that tend to get infected first, and that's partly just because it's more humid down there and the moisture sticks around longer. So we want to be as consistent as possible with the leaves that we're collecting. So traditionally, what we've done is waited until a leaf was fully expanded and relatively firm, but before it got very, very old. So the leaves where the arrows are pointing would be the leaves that we would aim for. So if they're too young and tender during the 2 to 3 weeks in those boxes, they tend to rot. So we wanted a little bit of firmness to them. So it's really important as well to grow the roses well. So that means good nutrition. Don't stress them out by dry stress, lack of water. So we need them to be actively growing. When they do get stressed out, get dry damage, um, we still can get black spot infections, but the rate of black spot growth is reduced. All right, so we bring those leaves up to the lab, lots of little baggies, things well labeled, and then we do a quick cleaning. So we found that just a little quick dip in 70% either isopropanol or ethanol and a little bit of a rub will get rid of any spider mites that might be there, and it leads to a little more consistent infection. So it abrades the epidermis, the cuticle layer just a little bit, and we find that we just have a little greater consistency. And then also getting rid of those spider mites, if there's any around, is a great benefit. And then we'll rinse them 3 times in distilled or reverse osmosis water, pat them dry, and the leaves are ready. So here's a classroom, and there's Ann Ganser, who helped me last year in the spring quite a bit. And so we'd prepare the boxes, have the labels on the lids all ready to go, put the right roses leaves in the right boxes, and we'd separate out on different tables which isolates or races that we'd be using. So we worked very hard to not cross-contaminate any of the different forms of the pathogen. So as we put out all the leaves, it's important to let the moisture that may still be on the leaves evaporate before we come later with the droplets of inoculum. So if there's big droplets of water left, the inoculum gets diluted, the liquid begins to run off the leaves. So I want to make sure that the free water is blotted off or evaporated off of the leaves. So now what I'd love to do is switch gears just a little bit and talk about the pathogen. We'll talk a little bit about how those physiological races are first characterized, how those isolates are obtained, and we ultimately get single spore isolates. So using known isolates that have virulence alleles that unlock or are able to infect specific roses gives us a great tool for continued research over time as we look for those RDR genes that confer resistance to some races of the pathogen, but not others. So typically what we do is we'll collect some black spot from some roses in the landscape off a specific cultivar, and we'll take those leaves back to the lab. We'll steep off some of those spores in some purified water, and then we'll do a dilute inoculation over some of the roses that we know are universal susceptibles. And then we get lesions just starting to form on our leaves. We'll surface sterilize those leaves, and then I'll put those little pieces of very, very young lesions on leaf tissue into typically PDA media. And the fungus will grow slowly. We see different colors in the different containers here. Different genetic backgrounds tend to look a little different. And then eventually we'll take some spores from these growing in Petri plates, and we'll do a really dilute inoculation over some more PDA plates until we get a little colony forming that we're confident likely came from a single spore. So we just have one genetic form of that fungus to work with from that point forward. And then what Vance did as part of his master's degree was characterize physiological races with these new isolates. And that ultimately helps us understand what's known as vertical resistance. So the fungus depicted by the circle on the left could have multiple virulence alleles. We could think of them as keys. They might just have one. And the rows, if it's a resistant rose with vertical resistance, we could think of the RDR genes as these locked doors and having them in a hallway. The pathogen has to open all the doors to get into that rose house to cause damage. All right, so there's a collection that we call the host differential. So the different roses depicted here on this table on the left that are genetically diverse. And then what Vance did was challenge all these roses with all the different isolates that he obtained. And then he eventually got isolates from Europe too. And then those isolates that have the same resistance by the negative symbol here or positive reaction with the plus sign, the isolates that have the same pattern across all these hosts get lumped together into the same physiological race. They can unlock the same roses and likely have the same combination or keys on their keychain. All right, so eventually we want to store these isolates to make them available in the future and to other researchers. So we found that the best way to store and work with them is to actually just grow them on rose leaves and get them away from PDA. And then we could put them in little cryotubes, dunk them into liquid nitrogen. They store perfectly. We've taken them out 10 years later and they woke up and didn't skip a beat. So when it comes time to prep the inoculum for some cultivars or breeding lines that we want to characterize, what we'll do is we'll take lesions from some leaves that we had in some of the boxes of our known isolate, and then we'll put them into little containers. We've been using just the sterile disposable 50 ml centrifuge tubes. We'll wash down the tables really well with alcohol, be real clean so we don't cross-contaminate our isolates. And we'll look for the lesions that don't look like there's any secondary infections of bacteria, add some purified water and gently shake it, dislodging some of those spores. And then within that liquid, there should be a lot of spores, and we'll avoid any leaves that begin to rot before this point. We'll put them in a cooler just to store them so they don't get to this point if possible. And then those spores, the conidia, these two-celled spores, are suspended within our water, and then we want to quantify the concentration and adjust the concentration of those spores for our inoculum. So using a hematometer, we'll see where we're at in that little vial. And so in the hematometer, we'll look for 1 millimeter by 1 millimeter squares. We'll count how many spores are in there. So the depth here is 0.1 millimeter, and the volume that's within this area here is 1/10,000th of a milliliter. So we'll count how many spores are in this square, and we'll do so over multiple squares, get an average, multiply by 10,000, and then we know how many spores there are per mil. And our target is 30 to 80,000 spores per mil, so 3 to 8 spores in this area. So this is highly concentrated. We dilute the sample to be within that. And then there's different ways to do the inoculations. So what we've been doing recently here for our just qualitative assays is to use a sterile pipetter and then just dab little droplets onto the leaflets. And it's best if we avoid the midrib, just because if a secondary pathogen, if a bacteria starts, it'll go through the leaflet more quickly. And then we'll put the lids on these containers, and then I'll put them in the lab. Over the years, we've done different inoculation techniques. We would use a mister and mist inoculum over the leaves. We'd waste a lot of inoculum that would roll off or go onto the paper towels. Here with that pipetter, it's a lot more controlled, and we've been happy with that for just qualitative assays. So the containers are best if stored in light. So we leave the lights on 24 hours a day. It keeps the leaves from degrading, keeps the leaves green, photosynthesizing. So there's enough time for that 2 to 3 week time frame for a black spot to start in a susceptible reaction. So one of the challenges that we found is that it's important not to have a temperature gradient. So here in the lab with the cubbies underneath, this is far away from the windows. The windows are on the left, the main hallway's to the right. This is the perfect little area here where we don't get the temperature fluctuations or gradient. If we have a warm lab counter from maybe heat pipes below the floor, that'll warm up the bottom of the container and then evaporate some of that moisture, and then the cool upper surface then would lead to condensation. So we want to avoid that. So be careful where you place these. In addition, if there's too much darkness, we ran out of space a little bit. So just for some race maintenance, some isolate maintenance. I stuffed a few down here just to see how they would do. And we could see that the leaves on the edge here that got more light were in good shape, but those that were a little too shaded degraded a little too quickly. After 2 days, what we'll do is we'll come back in and we'll blot off any of this inoculum, this liquid, because if we left it there, that'll lead to secondary pathogens, bacteria coming in. So we'll open up the containers, blot them off, stack them back up where they were, and then follow them for black spot development. So I love to check on them right after a week. We start seeing infections, and in very susceptible roses, we can get degradation very quickly and bacteria coming in. So we don't want to do an autopsy at day 14, hoping that we could determine if there was black spot or not, but we'll record black spot along the way here, typically following the boxes to day 14, and then we'll leave them even longer and just take a peek before we clean them just to make sure there isn't a very late infection. So eventually we could see which roses are resistant to the particular isolate and those that are susceptible. Some roses that have likely good quantitative resistance get a little iffy sometimes. So what we do then is look under a stereoscope and we'll check to see if there's a cervuli bursting through the surface. And that's our tipping point in terms of a clear susceptibility if we see a cervuli. But usually things are very cut and dry. All right, so we can use these assays not only for qualitative responses, but also quantitative. So years ago, we did a study where we misted little droplets over leaves and boxes. And then at a particular time point, day 12, we would measure multiple lesions, the largest lesions across all the leaves in the box, take an average, and we were able to compare lesion development. And what we have here in this figure is just the roses ranked by lesion size for each of the different races, and then significant groups for relative lesion size. We could also do other techniques, and Deanna's leading the way with that, where we put droplets on with a pipetter, so every droplet is the same volume, and then we take pictures of the box, and we come back later and look to see if a lesion developed where the droplet was, so we could look at ratio of droplets that lead to lesions, and then also measure leaf area, lesion diameter over time as well. So when we're done, we need to clean up these boxes really well so we don't cross-contaminate anything, rinse them out. I've been dipping them in alcohol. We used to wash them in bleach and then rinse them, and I've ruined a whole lot of clothes. So I've been dipping in alcohol here instead. And I did a big old clean up here a weekend or two ago, and hundreds of boxes, get them all prepped and ready with paper towels for the next reaction. So to leave you here with this presentation, I just love to share that this last spring and summer, we characterized 189 roses using 6 different isolates that represent 6 different races. And we found that 76 of them were susceptible to all the isolates, 17 were resistant to all 6, and then most of them had a mixture of susceptible resistant reactions. So there's likely some R genes that we picked up that that confer resistance to just some of those isolates, but not others. So this will become a really good validation group where we can eventually validate different markers that are linked to specific RDR genes. And Deanna will share that after lunch. So thank you very much. Are there any questions?
Okay. The question is whether the races are uniform in their virulence or this, I guess, aggressiveness.
That's a fantastic question, and this is what we found. Their aggressiveness ebbs and flows a little bit over time for a specific isolate. And so we're not sure exactly what always contributes to that. I suspect it's a combination of specific plant or leaf quality during the growing season as light is more intense, and exactly the nutrition management physiology of the plant. In addition, just the isolate over time seems to be a little slower and then suddenly seems to be a little more aggressive. But but we're grateful that at least over time with the host differential, with the isolate, that the patterns stay the same. So we know that there's a lot that goes into quantitative resistance, and a lot of it is environment, like you mentioned, Dave, in terms of locations, Overton versus College Station, and a lot of factors. So um, even though we want to breed for quantitative resistance, and it's really important, if we have a very conducive environment, like a real wet climate, um, these qualitative resistance, these RDR genes, will be very powerful and helpful. So there is variation for virulence. It's a little unpredictable as we're doing inoculation to inoculation. Um, and we're especially excited to contribute with the RDR genes.
Oh, Hawker was saying that have you screened or should you screen them, the isolates for microviruses?
Oh, that's a great question. So I know Ben Lockhart.
He's willing to work with you to get that done.
Oh, that even better. Perfect. Thank you, Maher.
So, no, Hawker, go ahead and email David with your questions and he can follow up with you.
Oh,
Yes.
that's Oscar. Okay.
Does this isolate lose variant if they are maintained in culture for a long time? And if so, how often does he pass them back through?
So do the isolates lose virulence if they're maintained in culture for a long time? And if so, how do you maintain them, or how many times can you pass them through?
That's a fantastic question, and that's something that we wanted to avoid even dealing with. So that's why we maintain them on roses now. So at first we're at single spore isolation. We'll grow them in culture, but then we didn't want to worry about that. So we know that they love roses. They'll tolerate potato dextrose agar. So yeah, so we just maintain them on roses.
Oscar? Oh, how do we know the races are not changing over time?
We don't.
Yeah, changing over time in terms of what we find in a garden or what we have that's growing in culture, changing where?
No, with respect to your collections, how do you know they're stable over time when you store them in liquid nitrogen?
Yeah, great question. So periodically what we'll do is we'll put them on some of the host differentials and just confirm that they're still stable. Actually, our concern usually is more so did we accidentally cross contaminate something, and for the most part, things have been pretty not cross contaminated and continue to behave as they've done in the past.